Welcome to this video on monohybrid cross and Mendelian principles of inheritance. In this session, I will focus on the overview of Gregor Mendel monohybrid cross, findings, interpretation, and derived laws and their significance. Let us begin with the overview of Gregor Mendel. Gregor Mendel was an Austrian monk and scientist. In fact, he was the first biologist who used mathematics to explain his finding. He was given a project to work to develop hybrid seeds by the monastery so that they can make good amount of money and do social service. However, his interest was to understand the inheritance, the mechanism of inheritance. He selected garden pea for his experiment, which is also known as Pisum sativa. So there were two objectives of his experiment. First, the objective of the monastery was to develop good quality seeds so that they can make money, and his interest was to understand the principles of inheritance. Based on his finding, he proposed laws which are applicable in predicting or which are fundamental to our understanding of the inheritance of genetic traits. During his lifetime, however, his contribution remained unrecognized and he died in obscurity. However, later on, when scientists revisited his experiment using different animal and model system, they were able to reproduce. They found also some variants which expanded the field of genetics, and therefore Gregor Mendel is now known as father of genetics. His experimental design was so brilliant, so intelligent that it looks like he addressed simple genetics in which recombination, crossover, linkage, sex, or environment do not seem to play any role on the expression of these traits or phenotype. Mendel addressed particulate hypothesis of inheritance. According to this hypothesis, traits are carried on discrete particles, units, or factors. At the time of Mendel, chromosome, DNA, genes, alleles were not discovered. Only information about DNA was that there is a material or chemical in the center of the cell or nucleus which is rich in phosphate. Because of this, he used the term factors. Factors retain their identity during transmission. They are not lost, they are not mixed up. And these factors in modern terminology nowadays are called as genes and alleles. This terminology was not used by Mendel. He used the term factors. Mendel's experimental design was brilliant and elegant. Therefore, I would like to go over the key features of his experimental design. He used Pisum sativa. Why he used? Because it grows faster and in large number. And as I said, he was the first biologist who used mathematics to explain his finding. Therefore, larger the number, more reliable were the results. Second, it is a bigamous plant, means both male and female reproductive organs are present in the same flower. This allows him to do reciprocal crosses. For example, the male gametes from the upper flower were dropped on the pistil of the lower bottom flower in this case, or alternatively the stamen or male gametes from the bottom flower were dropped on the pistil of the upper flower. Such crosses are known as reciprocal crosses, cross pollination, or cross fertilization, and this allowed him to study the effect of sex on the expression of phenotype in his experiment. He selected 7 unique traits. Unique means these were distinct and they did not interact with each other. That is why I used the term unique traits. Next point, very important point, he selected contrasting phenotype for each trait. For each trait, he selected contrasting phenotype. Means, for example, let's say height is a trait. He selected tall versus dwarf, contrasting. Color, purple versus white. Next important point of his experimental design is he used pure or true breed plants to start his experiment. It means that his P1 generation plants were always pure plant. What is pure plant or true breed plants? I will explain that in the next slide. So what are true breed or pure breed plants and how he obtained? He grew plants with a defined trait. For example, tall plant was grown for multiple times or generations, and if the phenotype was retained generation after generation, that plant was said to be or considered as true breed or pure breed plant. Now in modern terminology, pure breed or true breeds are known as homozygous plant. So in P1 generation he used homozygous plants. So let me discuss the traits and their contrasting phenotypes which he used in his experiment. He selected 7 which are listed here, such as stem height, pod shape and color, seed shape and color, flower position and color. And for each trait he selected pure contrasting phenotype. For example, height he selected tall and pure dwarf. For each trait he selected pure contrasting phenotype, and we conducted experiment using each pair. He found that in F1 generation only the phenotypes which are listed in the left column appeared, and they were called as dominant phenotypes, and while the phenotype which are listed here in the right columns, they did not appear and were called as recessive phenotypes. So as I said, he used 7 unique traits. They are unique because there was no interaction between these traits, and also there was no linkage. It means they were not on the same chromosome. So it will be interesting to know that there are only 7 chromosomes in pea plant. The karyotype of pea plant was not known at the time of Mendel, but now we know there are only 7 chromosomes. Therefore, these 7 traits appear to be on separate chromosomes. These are single chromosomes shown here. In the next coming up slide, I will make them double and will tell you the reason why they exist in pairs. After this background, I will present now monohybrid cross. I will define it, present the methodology used, findings obtained, and their interpretation and the laws derived from these findings. Then I will discuss Punnett Square method and medical relevance. So let us define monohybrid cross. This is a cross between two pure breeding plants with pure contrasting phenotypes of a single trait. That is why it is called as mono. Mono means single, that produces monohybrid offspring. For example, for trait height, he selected pure tall and pure dwarf, and for color trait, he selected pure purple and pure white. Such a cross was known as monohybrid cross. So let us discuss the methodology he used for his monohybrid cross. I will explain this using height as a trait. So in P1 generation, he crossed pure tall plant with pure dwarf plant, and the outcome in F1 generation were all tall plants. So the trait which appeared in F1 generation was known as dominant trait or dominant phenotype, and the one which did not appear, or in this case dwarf did not appear, was known as recessive trait or recessive phenotype. He experimented using other traits also and obtained the same result, and based on that he proposed the law of dominance or complete dominance. However, he was curious to know the fate of dwarf trait or recessive trait. Therefore, he raised the question, what happened to the dwarf or recessive trait? To address that, he self-crossed F1 plants in the next experiment, which I will explain in the next slide. To know the fate of the recessive trait in the next experiment or in this experiment, he self-crossed F1 plant which were monohybrid, and they were called as P2 generation plant, parental generation 2 plant. So P2 plants are not pure, they are monohybrid, obtained from the first experiment I explained already. In this experiment, he grew about 1000 plants, 787 were tall and 277 were dwarf. So dwarf is reappearing, it was not lost, it was not changed, it reappeared. They segregate and reappeared, and similar results he obtained using other traits as well. If you calculate the ratio of tall and dwarf or recessive trait versus dominant, it comes out to be 2.8 is to 1, which is rounded to 3 is to 1 ratio. So tall and dwarf ratio is 3 is to 1. These are rounded figure, and this is the probability. It doesn't mean that if we grew 4 plants, 3 will be dominant phenotype, 1 will be of recessive phenotype. It means 75% will be tall here in this experiment and 25% were recessive or dwarf. He conducted a similar experiment using other traits and he obtained a similar result, and based on that he proposed the law known as law of segregation. Now he was a he was he was interested to know the explanation or interpretation of this ratio 3 is to 1, and to address that he he used he applied simple mathematics law of probability to explain this ratio of 3 is to 1. So what is the interpretation of 3 is to 1 phenotypic ratio? I will explain that in the next slide. To interpret his finding of F2 generation, he used the following strategy. First, he made an assumption, and using that assumption, he devised a method of writing plant genotypes. So regarding his assumption, according to him, the 3 is to 1 ratio can only be explained if the factors are in pair. So his assumption was a trait is determined by a pair of factors, or in other words, you can say factors, genes, alleles, or chromosomes are in pair. This is universal truth, and you know that our chromosomes are in pair. So in the previous slide, I showed you 7 chromosomes in pea plant. They are not single, they are in pairs. So there are 7 pairs of chromosomes in pea plant. So using the assumption that a trait is determined by a pair of factors, he devised a way of writing genotype. So he selected a pair of letters for a trait. For example, let's say AA. You can choose any other letter as well, but in this example I will stick to A. So for dominant phenotype, he selected capital letters in pair. So capital A, and for the contrasting recessive phenotype, he used same letter but in small case, like small a, and using this strategy, he assigned the genotype of F1 plant in order to explain or interpret his finding. So using his assumption that the factors are in pair, he assigned the genotypes of F1 plants. So he started with writing the genotype of P1 plant which were pure. So P1 tall in this example, which was dominant, will be capital T, capital T. And for the contrasting dwarf or recessive phenotype P1 generation plant, the genotype will be same letter but in a small case, so small t, small t. So according to his assumption, the only possible genotype of F1 plant will be one copy from here, this parent, and one from this parents. So this is the only possible genotype, capital T, small t. So the F1 plant genotype is capital T, small t, or monohybrid you can say, or heterozygous. Using this genotype, he applied law of probability and can interpret his finding of 3 is to 1. In fact, all his laws have been derived using this assumption that the factors are in pair, or a trait is controlled or regulated or by a pair of factors. So he assigned genotype of F1 plant capital T, small t, capital T, small t, or monohybrid, and he interpreted the result of F2 generation using the laws of probability. The only possible genotype of the offspring from this cross in this case will be this capital T, black capital T can go with the red capital T or with red small t. This small black T can go with capital red or small red T. So these are the only four possible genotype in this situation, and these each is the product of two independent events. One is segregation and the other is fertilization, and each event has 50% chances. Therefore, overall probability of each genotype will be the product of their individual probability. So 50% times 50% will be 25%. So each has 25% chances in this case, and he interpreted that wherever there is one capital T, that will be tall or dominant phenotype, and where you have both small t or recessive trait, that will be dwarf. So he predicted or he calculated the F to G phenotype ratio to be 3:1, which is similar or same as reported by his experimentation. However, there is a catch here. These two are although tall plant, but tall phenotype or dominant phenotype, but they are hybrid or composite. Therefore, genotypic ratio will be 1:2:1. So one pure tall, two hybrid tall, and one pure dwarf. There are 2 points to note here. The dwarf phenotype or recessive phenotype always appear in homozygous condition. When both are recessive, you will see appearance of dwarf phenotype. Second, in human recessive condition, these hybrids are called as carriers. So what are the postulates and laws derived from the monohybrid cross findings? The first one is a trait is determined by a pair of factors. This is universal truth. I will not elaborate any further on this. The second was law of dominance or complete dominance. I will elaborate in the next slide. And the third is law of segregation. I will also explain this in the next slide. So let us elaborate on the law of dominance or law of complete dominance. First point is it applies to a hybrid condition. For example, here capital P, small p, only capital P will express, and the factor that expresses in a hybrid condition is known as a dominant factors, allele or gene, and the factor which does not express in a hybrid condition, such as P, small p in this case, is known as a recessive factor, allele or gene. The recessive factors expresses only in a pure or homozygous condition. So let us elaborate on law of segregation. Basically, this refers to separation of chromosome during meiosis cell division or meiotic cell division or reduction cell division, which happens during gametogenesis. So during gametogenesis, factors segregate so that each haploid gametes receives one or the other factor with equal likelihood. So gametes are formed during segregation, which are haploid in nature. They are on separate chromosome and therefore they segregate and give the result 3:1 ratio. Therefore, these factors are not located on the same chromosome or they are not linked. I will explain now Punnett Square method, its basis and application in human conditions. You have known that Mendel's used laws of probability to calculate or predict the offspring plant genotype and phenotype, which is a cumbersome and mathematical method. Therefore, an easy and quick method was devised by Reginald Punnett in 1905, 50 years after Mendel laid the foundation of inheritance. This method was used to predict the offspring genotype and phenotypes, and this is based essentially on the principles of Mendelian inheritance. For example, genes are in pairs, they segregate, they are not linked, and they show independent assortment. However, to utilize or to use this method, it is essential to know how to write genotype of gametes which are haploid in nature. Therefore, in the next slide, I will explain how to write the gamete genotype. It will help you to select appropriate number of the small squares and B to utilize or use it for human conditions. So I will now explain how to write gamete genotype using a trait which is shape of seed, round being dominant and wrinkled being recessive. So let's say you have a diploid genotype for round homozygous capital R capital R. In this situation you will make only one type of gametes which is dominant with 100% likelihood. The next is, let's say you have wrinkled seed shape which is homozygous small r. In this case you will make small r or recessive type of gametes with 100% likelihood. One type of gamete in both these conditions. The other situation is hybrid, let's say capital R small r, which is round in phenotype. In this case you will make two type of gametes, dominant and recessive, with 50% likelihood of each type. So in monohybrid cases, a maximum of two types of gametes are produced. You can calculate the number of gametes using formula 2 raised to the power n. So in monohybrid crosses or cases, you will make maximum two type of gametes. So two from one parents, two from another, 2 times 2 is 4. That is why you use maximum. In monohybrid cases, you use 4 small squares in the Punnett square method. So now I will explain how to use Punnett Square method. There are two requirements for using this method. The first one is you must be able to write the genotype of the gametes, and second you must be able to select small squares for appropriate number of small squares for the grid. For example, in this monohybrid cross, each parents can make two types of gametes, two from here, two from here. So 2 times 2 is 4. So make a grid having 4 small squares. Align the gametes from this parents vertically here and from this parents horizontally here. Put the combination in the corresponding square and predict the offspring phenotypic ratio, which is 3 is to 1, means 75% chances are of being dominant phenotype and 25% chances of being recessive phenotype. The offspring genotypic ratio will be 1 is to 2 is to 1, means 25% chances are of being dominant homozygous, 25% chances of being homozygous recessive, and 50% chances of being heterozygous. So you can see here in monohybrid cross, both the phenotypic and genotypic ratios are same as reported by Mendel using his laws of probability or mathematical approach. Therefore, once you know how to write gamete genotype and select appropriate number of squares for the grid, you can easily use this method. I will explain now the application of Punnett Square method in human conditions such as cystic fibrosis, phenylketonuria, and many more. These are monogenic recessive conditions, and in these conditions, Punnett Square method can be easily used. Let's say you have a cystic fibrosis hybrid or carrier mating. Cystic fibrosis is a Caucasian problem, genetic disease, which is very common in Caucasian population. So there are two carriers mating. From each carrier you will make two types of gametes. So make four small squares, align male gametes here, female gametes here, put the combination in the corresponding square and predict the phenotype. So 25% chances are of being normal homozygous, 25% chances are of being disease phenotype homozygous, and 50% chances are of being normal phenotype but hybrid or carrier. So phenotypic ratio will be 3 is to 1 and genotypic ratio will be 1 is to 2 is to 1. So 2 is carrier, 50% are carrier in this case. So this is commonly used in predicting the genotype and phenotype of offspring for a given individual case. However, if I ask you the frequency of the same disease in population, you cannot use Punnett square method. You will use another method which is known as Hardy-Weinberg equation. Hardy-Weinberg equation I will derive using this this data which is given in this square in the grid. So to calculate disease frequency in a population or in a country, Punnett Square method cannot be utilized. You use Hardy-Weinberg equation to calculate frequency of disease in a population, and that Hardy-Weinberg equation will be derived using Punnett Square data which I explained in the previous slide. This equation is commonly used in population genetics. So I will explain now derivation of Hardy-Weinberg equation utilizing the Punnett square data. So this is from a monohybrid cross. This is the possibility of genotype in an individual case. But if you look at the countryside or in a wide population, they will be these alleles will be mixed and randomized. So this is in the population. So let us calculate the frequency of dominant allele is 4 over 8. 4 are capital C, so dominant allele over the total number of alleles, which are 8. And the frequency of recessive allele will be small c is equal to 4 over 8. So frequency of both allele in that particular population will be capital C plus small c is equal to 1. This is equation 1, very important equation. We will use that to derive the Punnett square, the Hardy-Weinberg equation. So this tells you that the allele frequency in a population remains constant. This equation can be used to calculate the frequency of dominant as well as the recessive allele, and we will use that equation to derive Hardy-Weinberg equation in the next slide. So I will derive the Hardy-Weinberg equation using equation 1, which tells us that the allele frequency in a population remains constant, capital C plus small c is equal to 1, which is basically the and this is the Punnett square data. You will see that Hardy-Weinberg equation is another form of expression of the same data in the form of a quadratic equation which is known as Hardy-Weinberg equation. So this equation 1 is both sides are squared and giving you that this equation c square plus 2 capital C small c plus small c square is equal to 1. This is a quadratic equation. This is known as Hardy-Weinberg equation. This is another expression of the same Punnett square data. This capital C square is same as this one here, capital C small c. So if this tells you the frequency of normal genotype, this basically is same as c square. This is the frequency of disease. So if you are asked to calculate the disease frequency, you have to calculate the value of small c square, and these two are the carriers. So two parents, two carriers, 2 capital C small c is the carrier frequency. So if you are asked to calculate carrier frequency, you have to calculate the value of 2 capital C small c. You will not oversimplified this value. So this is how Hardy-Weinberg equation is derived. So the summary of Hardy-Weinberg equation: the frequencies of genotypes and alleles in a given population remain constant. It is derived using Punnett square data for monohybrid cross. It is used to calculate the frequency of disease, carrier, and alleles in a population. So, summary of monohybrid cross: a phenotype is determined by a pair of factors, or you can say factors are in pair or genes are in pair. These factors are also known as alleles or genes in modern terminology. In the hybrid condition, the factor which is expressed is known as dominant factor, and the one that is not expressed is known as a recessive factor. These factors segregate and retain their identity during gametogenesis. Overall notion from Mendel's finding and observation is one gene, one phenotype, means one gene regulates a distinct phenotype. So significance of Mendel's laws of inheritance or Mendel's finding: These findings are fundamentally important in our understanding of the inheritance of traits both in plants and animals. They establish the fundamental laws of inheritance such as law of dominance and law of segregation and law of independent assortment, which I will explain in the next video, and helps us to predict the offspring genotypes and phenotype, and forms the basis of modern genetics and genetic counseling. Based on that, we can apply preventive measures in certain cases to control and prevent spread of human diseases. Thank you very much for watching this video. Please like it, share it, and subscribe it, and let me know your feedback. Until next time, take care. Bye bye.